Good afternoon, ladies and gentlemen. First, I would like to thank the Dr. Rakim Mewal for her kind invitation to participate in this first ILBS Research Day, which I am sure will be successful. First, I will comment briefly on background findings we had in acutely decompensated cirrhosis, and this finding led us to analyze circulating immune cells and analyze blood metabolism in this context of acutely decompensated cirrhosis. Let me remind you the main results of the canonic study. We enrolled 1,343 patients admitted for acutely decompensated cirrhosis, referred to as AD. And among these patients, 70% had AD and no organ failure. This group was called AD no OF. This group was characterized by low-grade systemic inflammation and low 28-day mortality rate. 30% of patients had AD plus organ failures. This group was called ADOF. And in the West, we call this group ACLF. But here I will use the term ADOF. And this group is characterized by high-grade systemic inflammation and high 28-day mortality rate. In the canonic study, we assess systemic inflammation using very simple markers, leukocyte count and CRP levels. And as you can see, patients with ADOF were characterized by an intense systemic inflammation indicated by leukocytosis and high CRP levels. And because of leukocytosis, we wanted to know more about circulating immune cells in these patients. And therefore, we learned a lot from bioinformaticians who showed that analyzing genome-wide RNA expression in a whole blood provides outstanding information on immune cell subset in a single sample from a single individual. They use either microarray or RNA-seq, and in our case, we chose microarray instead of RNA-seq because microarray was cheaper. We enrolled 7 patients with advanced cirrhosis, 7 patients with AD no OF, and 70 patients with ADOF, including 8 patients with septic shock. In addition, we enrolled 7 LC subjects, and blood was collected in Tempus tube in every study participant. And in particular, these tubes were collected on patients' admission, therefore very close to admission. We performed genome-wide analysis of all blood RNA expression using microarray. Before commenting on blood transcriptome, I would like to show you some results we got in this study regarding clinical blood count. We use the following color code: orange for advanced cirrhosis, red for AD no OF, and violet for ADOF. In this study, we confirmed that patients with ADOF had leukocytosis, and we showed that leukocytosis was fueled by neutrophilia, which was significant. And in contrast, we also found that patients with ADOF had significant lymphopenia, which contrasts with the neutrophilia. Interestingly, there were no between-group difference regarding differential monocyte count. Then we analyzed blood transcriptome, and we performed 3 pairwise comparison: advanced cirrhosis versus LC subjects, AD no OF versus LC subjects, and ADOF versus LC subjects. The comparison used the same color code as previously shown for the clinical blood count. On the left, you have shown the Venn diagram analyzing differentially expressed genes. And as you can see, there were very few differentially expressed genes that were unique to advanced cirrhosis versus healthy subjects, or AD-NO-OF versus healthy subjects. In red, the upregulated genes; in blue, the downregulated genes. And in contrast, there was a large number of unique differentially expressed genes characterizing the comparison of AD-OF versus C subjects, indicating that a patient with AD-OF has a very marked altered change in blood transcriptome as compared to healthy subjects. And then we perform enrichment analysis of differentially expressed genes, and we started by upregulated genes, which are shown here in the middle of the slide. And as you can see, the pathways related to innate immune response, including TLR and inflammatory signaling, neutrophil degranulation, immune activation, generic cluster, were significantly enriched among upregulated genes in AD-OF versus healthy subjects, while in the two other comparisons, there was no significant enrichment of these signatures of innate immunity. These findings are consistent with the neutrophilia observed in the clinical blood count. Then we looked at, on the right, at the enrichment of downregulated genes, and we found that some pathways related to T cell activation, T cell signaling and activation, NK cells were significantly enriched among downregulated genes in AD-OF versus healthy subjects, indicating that there is a marked depletion in transcript related to T cells and NK cells, and these findings are consistent with the lymphopenia observed with AD in clinical blood count in patients with AD-OF. Interestingly, you can see that some pathways were also significantly enriched among downregulated genes in the comparison of AD-NO-OF versus healthy subjects, suggesting that the depletion in transcript related to T cells and NK cells preceded lymphopenia in acutely decompensated cirrhosis. This is an interesting finding that should be investigated in the future. Because neutrophilia is the fuel of leukocytosis in acutely decompensated cirrhosis with OF, we decided to characterize the phenotype of blood neutrophil in cirrhosis, and we enrolled 5 patients with advanced cirrhosis, 5 patients with AD-OF, and 5 patients with SC subjects. We didn't include here patients with AD-NO-OF because you remember the difference between AD-OF and healthy subjects was very, very little at the level of transcriptomic, and therefore we decided not to include this group. We isolated neutrophils in these patients, and we performed genome-wide analysis of neutrophil RNA expression using microarrays. On the top of this slide, I show 2 volcano plots for 2 pairwise comparison, advanced cirrhosis versus healthy subjects on the left, AD-OF versus healthy subject on the right. Each volcano plot shows red dots, which are upregulated genes, blue dots, which are downregulated genes, and the gray dots indicate genes that were not significantly different in the comparison. And as you can see, the number of differentially regulated genes up or down in advanced cirrhosis versus AC was very, very small, while in contrast, the number of differentially expressed genes up or down was extremely high in the comparison ADOF versus AC subject. Therefore, the transcriptomic profile of neutrophil in ADOF is markedly altered. Then we performed gene set enrichment of differentially expressed genes in ADOF versus AC subjects, and we found that downregulated pathways related to cell cycle, to migration, to TP53, the transcriptional regulation, were significantly enriched among downregulated genes in neutrophil from ADOF, and in contrast, pathways related to glucose metabolism, such as glycolysis, metabolism of carbohydrates, but also inflammation and neutrophil degranulation were significantly enriched among upregulated genes in the OD in ADOF, indicating that the phenotype of neutrophil in ADOF was extremely well determined and distinct from the phenotype in healthy subjects. So in summary, the landscape of circulating immune cell in ADOF is dichotomic. Indeed, there is leukocytosis, which is fueled by neutrophilia. In addition, this neutrophil exhibited increased neutrophil RNA signature related to granules and glycolysis. And in contrast to neutrophilia, we found a depletion of the lymphoid compartment involving mainly T cells and NK cells. And this is an important finding because probably the lymphopenia plays a very important role in the development of secondary infection, which are very common complication of decompensated cirrhosis with organ failure, and a very important cause of death in these patients. Because we found some interesting features of increased glycolysis in neutrophils in patients with ADOF, we decided to perform analysis of blood metabolome. And also we learned a lot from immunologists, and we spent some slides to give some concept coming from immunology. First, immunologists showed that acute systemic inflammation is an anabolic response which is energetically expensive because of synthesis of several inflammatory biomolecules such as cytokines, chemokines, lipids, etc. Because of increased biomass due to cell proliferation. Here we have neutrophilia, which is indicating stimulation of granulopoiesis in bone marrow. Also, because of cell migration and oxidative burst, and finally also because of activation of hepatic acute phase protein response. The paradigm of this response being the increased level of CRP. Therefore, you can assimilate the hepatocyte to the innate immune system in this concept. Taken together, these findings indicate that reallocation of nutrients, glucose, lipids, amino acids is required to fuel immune activation. This is the first point. Then we also learn from immunologists about glucose metabolism, first in naive, unactivated innate immune cells. As you can see here, glucose enters the cell. is used through glycolysis to give rise to ATP and pyruvate, and then give rise to acetyl-CoA, which is used by the Krebs cycle to produce an NADH, which is an electron donor to the electron transport chain, and therefore is involved in oxidative phosphorylation, which gives rise to 30 to 36 ATP molecules. It's also important to note that some pyruvate is used to give rise to lactate, and this reaction is used to replenish the NAD plus pool. And also a very important point is that glucose is used through the pentose phosphate pathway, we refer to as PPP, which give rise to NADPH, and also to ribose, which are building blocks for nucleotides. And also note that the Krebs cycle is fueled by fatty acid through acyl-CoA and giving rise to acetyl-CoA. Glutamine through glutamate fuel the Krebs cycle at the level of alpha-ketoglutarate. Therefore, this is the landscape in the naive innate immune cell. But when innate immune cells are activated, there is a reprogramming of glucose metabolism, in particular in neutrophil, where glucose is used to give rise through pentose phosphate pathway to NADPH, and also through to give rise to NADPH, which is it's very important cofactor for NADPH oxidase, which is the enzyme producing reactive oxygen species, which play a very important role in the production, in the very important antimicrobial role, of course. Very few pyruvate enter in the Krebs cycle, and also, as you can see, some intermediates of the Krebs cycle give rise to malate, and malate is used to give rise to pyruvate, and this is also used for producing NADPH for NADPH oxidase function. This is very important. Another point is that a large amount of pyruvate is used to give rise to lactate, and this process is called aerobic glycolysis. The final comment is that, as you can see, the Krebs cycle is broken because there are very few pyruvate is entering in the Krebs cycle, and also because Krebs cycle intermediate are used for malate synthesis, and therefore the oxidative phosphorylation doesn't work more and produce reactive oxygen species, which may play a role in the defense against microbes. What about beta oxidation of fatty acid during fasting in healthy subjects? And this cartoon shows the beta oxidation at the level of peripheral organs. As you can see, free fatty acid released by adipocytes are present in the blood, enter for example, the hepatocytes, is converted to acyl-CoA, and the acyl-CoA is converted to fatty acyl-carnitine to enter the mitochondria. And then fatty acyl-carnitine gives rise to acyl-CoA, which is a substrate for beta oxidation and ultimately to ATP production. Therefore, there is a system for delivering fatty acyl-carnitine to beta oxidation, which is very important. And in the context of systemic inflammation, such as sepsis, there is a massive downregulation of PPAR-dependent beta oxidation because, for example, there is a downregulation of the expression of the transporter of fatty acid carnitine, and there is a downregulation of every enzyme involved in beta oxidation, and therefore the ATP production is dramatically reduced in this context. And the only source of ketogenic body, ketone bodies, sorry, is the catabolism ketogenic amino acids. So these are important findings to have in mind to interpret our results obtained in our patients. We enrolled 831 patients with or without organ failure, 43 patients with compensated cirrhosis, and 29 healthy subjects. We perform high throughput metabolomics in samples collected in these patients. And interestingly, we found that in activated innate immune cells, we found features for glucose consumption through aerobic glycolysis to produce NADPH, amino acids and nucleotides. Therefore, to fuel an important anabolic response. We also found that features that amino acid released from skeleton muscle were used for synthesis in protein and nucleotides in activated innate immune cells. And finally, we found evidence that fatty acids released from adipocytes were used to produce bioactive inflammatory lipids in activated innate immune cells. Therefore, these findings indicate that in ADOF, nutrients are skewed towards activated innate immune cells in which there is intense anabolic metabolism. We also found features for an inhibition of mitochondrial fatty acid beta oxidation and ATP production in peripheral organs. This is an important point. And surprisingly, we found a very important inhibition of hepatic ketogenesis, and therefore we found evidence that the supply of ketone bodies, which are energy fuels to mitochondria in peripheral organs, was decreased. Together, these findings suggested a massive decrease in mitochondrial ATP production in peripheral organs, contributing to organ failure. This led us to a pathophysiologic model of ADOF. So at the beginning, you have a precipitating event, infection, acute liver injury due to alcohol or virus. This triggers the release of inflammation inducers such as PAMs and DAMs, which cause very severe systemic inflammation and oxidative stress. And then systemic inflammation may be a response involved in organ failure through three different pathways. First, by the stimulation of nitric oxide production in splanchnic arterioles, resulting in decreased arterial blood volume and subsequently to renal hypoperfusion, which is a major factor for AKI in these patients, and also systemic inflammation through cytokines can activate the migration of immune cells within tissues, and these immune cells can cause tissue damage, and this is also a factor of organ failure. And finally, we suggest that mitochondrial failure occur in peripheral organs, resulting in decrease ATP production and therefore energy production, a major mechanism for organ failure in AD-OF. So in conclusion, ladies and gentlemen, systemic inflammation for us is the driver of multi-organ failure in patients with acutely decompensated cirrhosis. Organ failure may be a result, at least in part, from the excessive anabolic metabolism in the activated innate immune system. And these findings suggest that manipulating immunometabolism may be another approach mitigating excessive inflammation or its consequence. I would say that probably we should reconsider how to use nutrients, I mean glucose, amino acid, fatty acid, for patient feeding in this context of acutely decompensated cirrhosis, and this will be a very important topic of research in the next future. I thank you for your attention, and thank you again for your invitation. It is always a great pleasure to be involved in IBS meeting, and I hope that I will participate soon in vivo to meetings in Delhi at the IBS, because I like very much this place. Thank you again. Bye-bye.